We recognize an object by its structure, shape, taste, and even smell, and eventually we respond to this input. Our cells, the building blocks of our body, are not different. Their response is highly plastic and can be instructed by the use of specialized materials, such as biomaterials, that mimic the environment of the human body. A fascinating concept called tissue engineering. Via this approach, BioMIMedics focuses on the regeneration of soft tissues like skin and blood vessels.
I think the biggest challenge in soft tissue engineering is to define materials that actually instruct cells to do what they have to do. So that really requires collaboration between engineers making the materials, designing the materials, and biologists understanding what those functions are and how you can coerce those cells to perform those functions in an optimal way in the body.
Within the multidisciplinary consortium of BioMIMedics, soft tissue engineering starts with polymer-based gel-like materials referred to as hydrogels. One fine example of the BioMIMedics portfolio is the phosphodiester-based hydrogel, developed at the IMO Institute of the University of Hasselt.
During the BioMIMedics project, we developed novel phosphodiester-based hydrogels. Their properties can be tuned to suit specific applications, such as cell scaffolding to grow new tissues, or as a wound covering material to treat difficult wounds like burns or bed sores. We hope that we've created an enabling technology that will improve patient comfort as well as healthcare in general.
A highly interesting feature of this phosphodiester-based hydrogel is its intrinsic property to respond to changes within an electrical field. This concept has been designed and executed at the Karim Institute of Maastricht University.
Electroresponsive hydrogels hold great potential for tissue engineering and drug delivery systems. The capability of these hydrogels to respond to an external electrical field make them excellent candidates to be utilized to improve cell behavior and production of extracellular matrix in tissue-engineered structures. In this project, we developed and evaluated a smart scaffold based on polyphosphodiester hydrogels. Conformational changes of this hydrogel induced by external electrical field showed improvement in cell matrix interaction and 3D tissue growth.
BioMIMedics hydrogel-based biomaterials hold great potentials, especially considering their broad range of possible applications and impact.
Well, the societal impact of this entire technology is extremely important, not only for medical applications, but also for many other applications. Maybe at some point robotic applications or food, for instance, where we are also interested in. So there are many applications, and that's why the societal impact is potentially huge. The academic participation in this research is important because that's where a lot of ideas are coming from, and it's also easier to create a multidisciplinary environment just by collaboration between institutions, and obviously collaboration with companies that eventually make this happen and commercialize, and in an early stage think about how can we scale this up and commercialize.
To realize commercialization, BioMIMedics bundles its forces with SMEs in the EU region. The young startup company Corium, focusing on creating bioengineered alternatives for the leather industry, is an eminent example.
The reason that we appreciate the cooperation with the Maastricht University is that it allows us to work on our dream to make a revolutionary product for the leather industry, and the cooperation is really perfect because the people we are working with are really dedicated and top scientists.
The opportunities are evident. A bright future for smart hydrogel R&D starts today.